Typically, biosamples have been stored in the frozen state with the belief that freezing is the only way to provide guaranteed stabilization for nucleic acids. However, this method is an inefficient use of energy resources. It is generally manual, consuming large amounts of labor to maintain and retrieve samples. Further, freezers take up quite a bit of space just for the refrigeration equipment. GenVault has created a better way to store and transport samples in the dry state at room temperature. The GenPlate is only one part of the solution. The GenVault system is automation friendly and labor saving. It is high density, saving space, and it does not require refrigeration, which saves energy. The GenPlate works by incorporating FDA paper, which is a proven technology that has been used extensively in forensics and agriculture for 18 years to preserve DNA containing samples at room temperature. The paper works by lysing cells upon contact, inactivating viruses and bacteria. These features enable the GenPlate to be shipped at ambient temperature with no biohazard labeling whilst maintaining DNA quality. The GenPlate is simple to use. Just add 10 microliters of your sample directly to each well containing paper in the plate. The plate can be easily dried in a desiccated environment or in one of GenVault's drying apparatus. This is sealed and stored until the aliquots are ready to use. The GenPlate is available in a variety of configurations, each suited to different users' needs and sample types. For example, a stick tube of blood can be dispensed into a single region GenPlate for whole blood containing 384 aliquots. Alternatively, those desiring a higher storage density with fewer aliquots can spot the blood sample onto a 3-region plate with 96 aliquots per sample and 3 unique samples onto a single plate. Our 6-region plate is optimized for the storage of the white blood cell fraction, boosting per aliquot yields and density. Our 12-region and 24-region plates are best suited for smaller samples and for backup of primary sample collections. These GenPlate configurations are available for use with other types of samples as well, such as buffy coat, purified DNA, plasmids, bacterial stocks, buccal samples, etc. Sample sources from humans as well as samples from diverse animals and plant life may be stored on GenPlates. There are 2 general types of GenPlates. One is FDA paper for raw or unpurified biosamples, such as whole blood, buffy coat, buccal samples, bacterial stocks, etc. The other type of GenPlate is FDA paper with an additive for purified DNA and plasmids. The additive assists in making more efficient recovery of purified DNA from the paper. Without the additive, the purified DNA is too tightly wound around the matrix of the FDA paper to be recovered effectively. Purified DNA on a GenPlate has a recovery rate of 70%. Plasmids on GenPlates have a recovery rate of approximately 80 to 90%. The GenPlates for unpurified biosamples are sealed in silver foil. The GenPlates for purified DNA and plasmids are sealed in blue foil. Purified DNA and plasmids must not be stored on GenPlates for unpurified samples. When you are ready to use your sample, just punch the aliquots out of the GenPlate and use GenSol to recover high-quality double-stranded DNA suitable for a multitude of genetic assays, including PCR, genotyping, sequencing, and whole genome amplification.